So my great pleasure to join you for this seminar. All right. So this is the outline for today's talk. First, I will spend about 10 minutes or so to talk about introduction and the background for the 3D genome structure. Then I will briefly introduce the current method and the technologies and how we use to study 3D genome. And then, well, the lecture will move on to the relationship between 3D genome and gene regulation. And finally, I will introduce you some tools and resources for you to explore 3D genome data and how you can use them for your own research. Okay, so again, so this is a lecture rather than a conference presentation. I really hope if you have any questions, you can stop me anytime and we can turn this into more like a roundtable discussion rather than I tell you everything I know about the 3D genome. All right. So I will start my lecture with this quote, hope it's famous quote. Life is like a bowl of noodles, right? Not a box of chocolate, right? So in this case, what do we mean by noodles? Of course, noodles are DNA, and the bowl here is the nuclear envelope. You can see different colors. The noodles means there are different chromosomes. So you can tell that different chromosomes have their unique space inside cell nucleus. So these are so-called chromosomal territories. Let me change it to laser pointer. Fantastic. All right. And then when you further zoom in, you can begin to see the human genome is organized into different compartments, something that we are really familiar with, the euchromatin and the heterochromatin. So inside of each different compartment, there's some kind of the hairball-like structure. So we will spend a lot of today's lecture on those hairballs, in which we term TADs, or topologically associated domains. And when you further zoom in, this is the highest resolution. You'll be able to see the chromatin looping events that we are more familiar with. So chromatin looping is more directly related with the gene regulation, as I'm going to demonstrate in the next slide. Okay. So this is really just the basic diagram for gene regulation in the mammalian genomes, right? So in the center, we have a promoter sequence whose role is to recruit RNA polymerase and initiate gene transcription. So far away from the gene promoter, there's an element called the enhancers, whose main role is to recruit tissue-specific or developmental stage-specific TFs. Right? So I believe the enhancer concept has been covered in your previous lectures. For example, I'm sure you have learned how to use ChIP-seq for his modifications, or use ATAC-seq to identify enhancers in the human genome, right? So although enhancers can be located far away from the gene they regulate, so they do play a critical role in gene regulation. This is one of the most famous example. In this case, this enhancer is located more than 1 million base pair away from the gene they regulate. And actually this enhancer is located inside intron of another gene, right? But when the scientists do experiments, when they delete this distal enhancer, there's a severe phenotype. Right, so there are two mice. You can tell which one is good, which one is bad luck. Right. So in this case, the guy on the left is wild type. Seems very well fed, a little bit chubby, right, but still looks very furry and cute. The guy on the right, this unfortunately is a mutant. So the enhancer sequence is deleted, and no exon is affected. But still, you can see the limb for the mouse is severely underdeveloped. So there are quite a few concrete examples like this to show the effect of enhancer manipulations, right? Again, I think these slides probably have been covered by Shirley before, but I just want to talk about it a little bit more to make sure we are on the same page, right? So far, there has been thousands of GWAS studies has performed in the human genome. For example, there might be more than 5,000 or 6,000 SNPs. They are related with different human disease, such as cardiovascular disease, or even cancer, or different human traits, right? But one of the surprising findings from the GWAS study is actually over 93% of these these SNPs, this disease or trait-related SNPs, they are located in the non-coding part of the genome. Okay, and then another study showed that actually majority of these GWAS SNPs, they are located in DHS sites, which means potentially means open chromatin regions in the genome, right? And then the idea is actually if there's some mutation happened in the distal SNPs through prompt looping, these defect enhancers might contribute to the misregulation of the target gene through prompt looping events, right? More recently, people began to show that even without mutation on the enhancer itself, right, when you only disrupt the 3D genome structure, you begin to severe phenotype in the human body. In this case, this is in the wild type. There are two chromatin domains. In this case, the domain 1, they have this green band is enhancer sequence. And in the domain 2, there's oncogene. So normally, because each element have their unique space, so enhancers don't really talk with the oncogene. But when the patient have the IDH mutation, in this case, they will lead to the aberrant demethylation patterns. In this case, as we will cover later in this lecture, that to maintain such chromatin structure, there's a protein called CTCF need to bind to the DNA and some sort of like seal the chromatin domain. But once you have the IDH mutation, the DNA binding motif, CTCF motif, is methylated. So as a result, the protein can no longer bind to their motif. As a result, the 3D structure is disrupted. And then the enhancers can now talk or contact the oncogenes and further drive its expression up by almost fourfold. As a result, it contributes to cancer-genesis. So here is another example. In this case, this is actually a SV or structural variant lead to the change of 3D genome structure. Okay, so in the normal chromatin 3 in the human blood, right, there's an enhancer regulating GATA2 through chromatin looping events. So GATA2 is expressed, and this oncogene EVI1 is silenced, which is good. But a main category for AML leukemia patients, there's a inversion 3. Is this big chunk is inverted, but no DNA sequence or mutation happened in the enhancer. But because there's an inversion happened, so this enhancer now is shuffled pretty close to the EVI1 and form chromatin looping events with the EVI1 and for the drive up expression for this oncogene expression and for the contribute to leukemogenesis. So I want to say that this is actually the so-called enhancer hijacking, and the enhancer hijacking concept began to gain popularity only in the past few years. And I think this will be a really, really exciting research directions for the cancer studies. Right? Okay, so next we are going to talk about the technologies or method to study 3D genome structures. So of course, ideally, if we can really see everything with our own eye, that would be ideal, right? And we have made some progress in that end. So as mentioned in the beginning, there's a chromosomal territories in the genome. So this paper from 2005, in this work, scientists began to label each chromosome with different color of dye. And when they combine everything together, you can begin to see these beautiful chromosomal territories inside the human cells. So you can see chromosome 1, 18, 20, and then you can see there's one copy of chromosome 18 here, and here's another copy of chromosome 18, right? So in the human genome, seems that these two alleles, they are not located together. Right? We can further zoom in, and then a very common technologies used to study chromatin interactions is called FISH, stands for fluorescent in situ hybridization. So we can use different colors, even 2, 3, or 4 colors to do FISH. So the idea is like this. Suppose you have two regions in the genome, say in this case L2 and L1. You want to investigate whether there's chromatin looping events between L1 and L2, right? So all you need to do is you just use different color labels to label that strand of DNA. You just need to denature the double-stranded DNA and put the label there, right? And your control is usually, because you are investigating L2, usually you go to the other direction and you label the L3 with another color. So the idea is the genomic distance between L1 to L2 and L3 to L2 is relatively the same, right? And then you do a FISH, and then you observe them under the microscopy. So in this case, you can see actually in this case, the red and green, these two dots right? They are located close to each other, meaning they are really close in the 3D space. Right? But this sort of light bluish color, although they have the same genomic distance, is located pretty far away from the red green spots, meaning the prompt interaction or the looping is only between the L1 to LT. And then there's no, and L1, L2, and L3, they're pretty far away from each other. So one potential limitation for a fish is actually you really need to know which element you are investigating. Right? So in this case, because you need to know the location for L1, L2, so that you can design your probes. Right? So the other thing you also need to know is fish is still relatively low throughput. Each time you only investigate one pair of loci, and then the resolution is also not ideal, usually tens or even hundreds of kbs, right? Okay. So about this 2002 science paper, Dr. Joop Dekker invented this technology called the 3C, which stands for chromosomal conformation capture. So this has really created a new field in the study of 3D genome structures. Right? Actually, now we're looking back, the design is quite simple and straightforward, but I think this is really groundbreaking, right? So inside your cell nucleus, suppose this is strand of DNAs. So although they are located far away from each other, they are captured by this protein. So they are close in the 3D space. So what you need to do is you cut the DNA with restriction enzymes. They can be 6 base cutters or 4 base cutters. And then sometimes they have this sticky end, not evenly cut, right? So you cut the DNA, you will have product like this, right? And then in the next step, you just need to induce ligations so that this part is ligated to this part. And in the last step, you do the reverse cross-link, remove the proteins, and you have a product like this. So ideally, this is from a different part of genome. And all you need to do, you just need to design primers and then do qPCR, have these interaction frequencies. Right? So Hi-C resolution, as you can tell, because you design primers to do a qPCR, is really base pair resolution, right? Give you high resolution results. But again, the potential limitation is for this is, again, you need to know the region you are testing, right? But sometimes you do. In this paper, Nature 2012, is back then when I was a postdoc in Bing's group. In that paper, we were able to identify or predict there's an enhancer on the right side. They are enriched for histone modification, H3K27 methylation, right? We predict this enhancer regulate this gene on the far left, so across almost like 200 kb, right? And we suspect this enhancer regulate this gene only in cortex, in the mouse cortex. Therefore, my collaborator Yin actually she did a 3C experiment in both the cortex and the brain. So when you do the 3C experiment, you usually design a series of primers surrounding your targets. So on the right hand, this is your anchor point. On the left hand, this is the walking primer, right? You really have 5 or more primers. And this sharp peak right above the gene promoter, suggesting that there's interactions between this promoter to this anchor point. Right? So this is a 3C experiment. So 3C is still widely used in different in current research. Okay? And then the really groundbreaking discovery is made from same group, Joop Dekker's group. He in 2009, right? I think he collaborated with Eric Lander's group. They were able to greatly expand the 3C experiment into called Hi-C, which is really high throughput. So in one experiment, they can give you the genome-wide chromatin interactions. And because it's taking advantage of the sequencing platform. Right? So in the beginning, you can see this plot is very similar to the 3C experiment, right? You cross-link your DNA, you cut them with the restriction enzymes. The only difference here between 3C and Hi-C is actually here, when you fill the end, you add a biotin, right? And then the following step is pretty much the same. You ligate your DNA and reverse crossing and then share DNA, bring them into fragments. Only here, because when fixing your end, you add biotin. In this case, you can use antibody. You pull down the fragments containing the biotin, and then you can sequence libraries that only contain the biotin fragments, right? And then you are hoping that because when you do a sequencing, you use pair-end sequencing. You have one end coming from one part of genome, and the other end come from the other part of genome. And then this is, once you have the sequence results, potentially millions of reads or even billions of reads, you can begin to construct the data matrix. Okay. Again, when you process in the ChIP-seq or ATAC-seq data, your data is one-dimensional. That means actually you can potentially even have base pair resolution is one dimension. But when we talk about the Hi-C, you are talking about two loci interaction with each other. So it's a n square, right? In that case, human genome is 3 billion base pair. You cannot do the base pair crop interactions. That would be 3 billion times 3 billion, right? So the result, we usually, when you process Hi-C data, you put data into different bins. Of course, the deeper sequencing depth you have, you can use a smaller window, right? And then as a result, so suppose this is your 1 KB window, this chromosome 1, 0 to 1K, 1K to 2K, blah, blah, blah. And then your y-axis is the same thing, right? You're interacting, you are investigating the chromosome interactions within chromosome 1, right? So each data value here, for example, 7. 7 means there are 3 reads. One end mapped to the genome loci 0 to 1K, and the other region mapped to the region 3 to 4K, right? And then in this case, here, this spot, you have 10 reads, one end mapped to 1K to 2K, the other end mapped to 6K to 7K, right? So you can tell, right, the stronger value or bigger number you have, potentially there's strong chromosome interactions between this loci to the other loci. So we're good? Good. If there's no questions, I'll keep continuing, right? All right, now you can convert this data matrix into the beautiful heat map that you use to see, right? Just given different shade of red or purple, whatever your favorite color, right? So in this case, you can see in case the strong red dot here means the same thing. There's chromosome interaction between this loci to this loci, right? You may notice that this is we're investigating chromosome 1 to chromosome 1. So that's why the data is symmetric, right? So this is the first figure that draw in this Hi-C paper, Science 2009. So what they are showing here is the chromosome interaction for the whole chromosome 14, right? So immediately you can begin to see there's a clear patterns, right? A lot of the data analysis pipelines or algorithms is really trying to dissect the Hi-C data matrix and looking for patterns, right? So this is one way to representing Hi-C matrix as a square, but sometimes you can also see the triangle-like shape. So what did it do, right? So essentially you can imagine just this square, you can rotate it to 45 degrees, have something like this. And because Hi-C data is symmetric, so now you can only see the half, right? So essentially this square and this triangle, they're essentially the same. Just people have different preferences. Some people are more familiar with this triangle-like structures. The only difference here now is each dot in the Hi-C mapping triangle indicating interactions between this loci and this loci, right? Has nothing to do with the loci right below it, right? So initially I take this as for granted, and then later on some people ask me is what's each dot stand for? Okay, in this triangle-like structure, dot here means the interaction between this region and this region. And you can begin to add your favorite genome browser shots here, right? So you can directly linking the genes or your his modification ChIP-seqs with chromosome interactions on the genome. All right. So one feature you can observe is actually the closer you are to the diagonal, right? The values are higher, right? So in this case, the plot, the x-axis is the genomic distance between the two loci, and the y is the contact probability. So essentially that means the closer the two genomic loci to each other, they tend to have chromosome interactions, which is relatively intuitive, right? So imagine, so this is your DNA, right? The idea is when the two parts are close to each other in genomic distance, it's easier for them to form a loop and contact each other. But at this long end, here to here, you need to form this much bigger loop, right? So the chance for this is really smaller. So this is what we call a distance decay in the Hi-C data sets. Okay. Okay. So even if this is the first time you see a Hi-C map, you can observe clear patterns, right? So what you see here, you can see clear triangles, right? So this is actually back then when I was still a postdoc in Bing's group. We spent a lot of time trying to write computer algorithms to identify such triangles in the genome. I think since our paper published in Nature 2012, so far there has been at least more than 20 to 30 different algorithms to identify the accurate locations for such triangles. Right? Sometimes you can see there's a big triangle and there's a smaller triangle. They call these subtags. Right? So what do we mean by tags? What we think as potentially each tag is potentially, this is your DNA, it's potentially one of these hairball-like structure. So within each tag, the genomes are more frequently contacting each other, right? So this is another test. So you can see these two hairballs, they are spatially separated from each other, right? Therefore, potentially there's important biological indications when you form a tag, okay? So how do we identify tags? Again, as I mentioned, so far there has been many tags identifying algorithms. Maybe if you're interested, you can also write your own. But in this lecture, I'll just briefly introduce the algorithm back then we developed. I think it's still commonly used in this field. Okay, so this is what we call a directionality index. I think potentially in your homework, I think it's part of your homework. All right. So again, because we cut the human genome into different bins of the equal size. Right? Suppose you have 100 reads, right? Because we are talking about a pair and the reads. By expectation, you would expect if this is one end of reads, this is a bin, you expect the other side of the reads, half of them will go upstream, half of them will go downstream. So roughly your expectation will be 50-50, right? But what we observe in the Hi-C real, in a real Hi-C map, we sometimes see like 90% of the reads, or even a big portion of them, majority of the other end of reads will be aligned to downstream rather than they'll go to upstream. So that means there's actually a bias. So this part of the genome, they tend to interact with the downstream rather than upstream, right? I hope we are clear on this point. And then we can actually compute simply put, you can just construct a 2x2 values, right? You are expected over observed. And then you can compute a p-value for each bin. In this case, each bar is one bin. You can compute this probability, it tend to interact with downstream or upstream, right? So the beauty for this works actually once you align this directionality index. So this is downstream, this is upstream. You can see they align really perfectly with the triangle, right? And conceptually, this is also very simple. You just have the interaction bias, and then you can just write a HMM model to call the tags. Right? So this is what we did back then, and this is what you will do in this week's homework. Okay. So one surprising discovery we made back then, and people keep making similar observation, is actually tags. They are really stable in different cell types. Right? It's quite surprising. So again, in this case, there's 5 different cell types: stem cell and a different cell lineage differentiated from the stem cell. And you can see this is the Hi-C signals. Right? You can hardly tell the difference for the Hi-C map from top to the bottom track. Right? Big triangle, smaller triangle, big triangle, big triangle. And later on, in one of the work in 2016 from Bing's group, they even show the tags across 20 different human tissues. Again, tags annotation is relatively stable, quite surprising. This just gave you a more quantitative view, right? So before, in the previous slides, we just gave you the raw Hi-C signals. And this is the directionality index. So you can see, surprisingly, the transition between tags is really sharp and conserved. This is a big tags we have seen in the previous slide. This is the small tags, right? Quite surprising. So you may ask, tags is so conserved in different cell types, and they might have some potential functions, right? So we will talk about their functions later. So another idea is actually, since they are conserved in different cell types, how about species? So in the same paper, we compare the tags annotation between human and mouse. Okay, so this is a syntactic region. between a human and mouse. Okay. In this case, you can see this is CCDC41, CCDC41, right? And then BTG1, BTG1. So they are really syntactic regions. In this region, you can see there are 4 big tags, right? So they are really conserved between between human and mouse. Actually, we also published a paper in Nature last year as well. There are certain regions you can even link the conserved tags between human to zebrafish, despite hundreds of millions of years of evolution. Right? It's quite surprising. Okay, so tags are important. What's the potential phenotype if you disrupt them? So this is actually the discovery from Manowitz group in Cell 2015. So they actually have this group, Manowitz group, have published multiple really great paper on function for tags. Right. So this is just one of them. In the top panel, again, you can see this is a tags annotated in human genome. There are 3 big ones in this region. There's a PAC3 gene and an EPHA4, right? So you can see what they observe actually when there's actually a deletion or inversion or the duplication. So all different type of structural variations, they all disrupted the TAG boundary. So as a result, the TAG structure is disrupted. So there's a severe phenotype you can see in human biology. So when they performed an experiment in the mouse, they were able to recapitulate the human disease in the mouse as well, further to confirm the importance of tags. Okay. Again, so this is a very nice review written by my former lab mate, Jesse Dixon. So in this review, they essentially go over a lot of publications. So these are the potential functions for tags, right? In the first scenario, potentially the genes located in the same tags, they are potentially co-regulated. And the idea is that because the enhancer located in the same tags, they can regulate the same set of genes because they are spatial proximity. Similarly, if an enhancer is located in tag 2, then it's very hard for this enhancer to get out of this hairball and regulate a gene located in the other tags, right? And then only in human disease we begin to see when the tag boundary is deleted, enhancers can regulate wrong target genes. So this is the so-called enhancer hijacking. So the other idea is that there's the tag boundaries can further stop the expansion of the repressive chromatins or the spread of transcription as well. So again, you can see the tag boundary is really just as a boundary to stop the expansion of either repression or active gene transcription. Right? Okay, so if there's no questions, I'll continue to the rest of the lecture. So we have shown that potentially the tags are really important, especially the tag boundaries, right? So what are enriched tag boundaries? So in this case, this is actually a enrichment analysis we performed back in the Nature 2012 paper. These are the histone modifications you might be familiar with in your previous lecture. So again, we see this strong enrichment for H3K4 trimethylation, which is active promoter mark. We also see the enrichment of TSS and then the enrichment for GRO-seq, which stands for active transcription, right? Another interesting element that we see is this guy, the sign element, which is a repetitive element, right? I remember, if I remember correctly, my good friend Ting Wang also gave a lecture in this class, right? So he's expert on the repetitive elements. It seems actually potentially retrotransposons also play a critical role in the formation of 3D genome structure as well. So besides this transcription and sign element, we observe the enrichment of this guy called CTCF, right? So CTCF is an important protein, right? Initially, it was discovered like 20, 30 years ago. Initially, it was discovered as a repressor. Actually, later on, people began to show CTCF can potentially act as an enhancer. Hold on 1 second. There's finally I see a question. Is there a question?
You can leave the question to the end of this recording. Does that work or?
Yeah, I think this is a simple question we can address. Yeah, so the question here is now all the tags are pretty close to each other. So whether potentially there's might be a gap between the two adjacent tags. I think the answer is yes. Because in all the previous paper, we usually see how much the genome is covered by tags. Usually it's between 80 to 90%, right? So potentially a lot of the genome, they are just not organized in those hairball-like structures. So tags, they don't necessarily located immediately to next to each other. Right? Hope that I address your question. All right. Again, so back then we discovered that the CTCF binding is strongly enriched at TAT boundary. So this is TAT boundary. And then this paper actually is from our paper last year. That paper is about the genome annotation and the Hi-C data in zebrafish. Right? So we can see even in zebrafish, CTCF binding sites, they are still enriched at TAT boundary regions. And similar observations have been made in human, mouse, and zebrafish, and quite a few other species. So it seems that the CTCF could play a critical role in the TAT formation, right? So how did it happen? So this is a really a more accepted concept right now. It has been proposed that loop exclusion has been really the foundation for the formation of tags. Right? So previously we showed that the CTCF is enriched at the tag boundary. So each of these arrow is a CTCF because we are talking about the proteins, they bind their motifs. So motifs have their directions, right? Either downstream or upstream. Right? And in this case, what the people have shown actually is the coordination between CTCF and another complex, which is called a cohesion ring, work together to form the task. Right? So this is a cohesion ring. You can see it has a SMC1, SMC3, and a RET21. Right? So they can form this ring type of like structure. So this ring will fall on the genome from this ring, and then the genome will go through the ring, driven by different potentially. There's a observation is a PAL2 directed. So the genome is being propelled by outside force. They keep going through this ring like this, going through the ring, and then they began to see CTCF. So when the CTCF see the cohesion ring, it will stop. And but the idea is that the two CTCF, they need to have a convergent motif. They can stop. If they are on the same direction, or they are both go outside, outwards, it won't stop. So back then, when myself first see this observation, I was like really surprised. So how is it possible? How the genome is so smart? They can only observe like genome is like this. And then in the same year, there are three back-to-back high-profile papers, Nature, Cell. They make the similar observation. And then in one of the paper, I believe they even using CRISPR to invert the direction of CTCF. They didn't change anything. All they did is invert the direction of CTCF binding sites. The loop can no longer form. So it's quite amazing, right? Again, to loop exclusion has been proposed to be the mechanism behind a TAT formation, and the two CTCF anchor, they need to be convergent, right? And actually, there has been shown that over 90% of the quantum loop events, actually, they have convergent CTCF binding sites. All right. Cohesion and CTCF, they're really required for the functional task. So in this paper, Rao et al., so the last author is Lieberman, who is the first author for the Hi-C Science paper 2009. So their group uses the background system. They can quickly dissolve the RET21, which is a part of the cohesion complex. So when they remove the cohesion complex, this is before the removal. You can see there's a very nice Hi-C map. This dot is the quantum looping events. So once they removed a cohesion complex, you can see the Hi-C map is essentially, I don't know if you can say destroyed. You don't see those patterns at all. And then because it's diagram system, they can begin to induce back the cohesion complex. And then they can begin to see this loop is formed again. So the cohesion is really required for the formation of quantum looping events. Okay. As beginning, I have mentioned that ideally, you need to see everything with your own eye. Right? So if in the future that can be done, that's really the best. But still, right now, the genomics is really the much easier to do because you don't really need a lot of special equipment. But imaging, especially storm or chromosome painting, has really made a stride in recent years. So this paper is from Alistair from Stanford group. So in this case, they were able to label, you can see this strand of DNA. They can label this strand of DNA with different colors, red, orange, and purple, right? So they use a series of hybridizations. And as a result, you can see your labeling with a super resolution microscopy, right? Like this, right? So what they'll see in their imaging, right? So they can still see this type of TAT-like structure from a super resolution microscopy data, right? So one thing is once they merge the observations from thousands of cells, they can see things like this. So one thing I'm not sure if I have time is actually when you observe TAS, these are from different cells. You can see it's the majority of the regions, they agree with each other, but potentially there are still cell-to-cell variations. I think that would be falling into the single-cell analysis approaches. Okay, so the other thing is, so far we talked about genomics, right? In those Hi-C matrix, we say, okay, 20 is, means there is a stronger complete action. 100 is even better, 0 is no interaction. But we really trust this data because in the end, you imagine this inside the cell, you are talking 3D genomic distance, but what the data in your hands, they are really just genomics, right? Do we trust the genomics data? So the image data actually from this paper really give us confidence moving on. So again, in this figure, the y-axis is Hi-C number of reads, right? From zero to 1,000, right? The Hi-C read intensity. The x is spatial distance observed from the super resolution microscopy. So you can see they almost have this perfect anti-correlation, which is great, right? It means the bigger the number in the Hi-C data matrix, the smaller spatial space inside the cell nucleus, right? They really give us a confidence that indeed you can use Hi-C data to make further inference for the 3D genome structure inside the cell nucleus. Okay, we have spent a lot of time talking about the tags, and we hinted something about the gene regulation. In the next maybe 20 minutes or so, we'll continue that line. Right? Sometimes it's really hard to distinguish the technologies and observations and their impact on the gene regulation. I'm trying to do my best to separate the lecture, but next one, again, they are pretty closely related. Okay, so this figure is from the original Science 2009 paper. Again, this is the chromosome-wise interactions, 14, right? Again, each red spot meaning there's strong chromosome interactions between this region to the other region. To make things look better, right? So essentially for each diagonal, you can compute a average, right? So you can see the expected value for here will be really small. The expected value for here will be really big or strong, right? So figure B, essentially they just use a observed value divided expected value. So, and then if you take a log, you can see the more clear difference between positive and negative, right? You can have a red and blue. Figure C is really interesting. You can see the pattern is much more clear, right? So what they did is they compute for each cell, not the real cell, each cell on the data matrix. They can compute Cij. You can compute the correlation between the ith row and the jth column. Right? For this spot, same thing, you compute the ith row and the jth column. So what does this correlation really tells you, right? So essentially, if the two cells are close to each other, it's not surprising their interaction profile across the whole chromosome are similar. So these two are red. But what is really surprising is actually this spot. That means the location here and the location here. Although they are pretty close, they are far away in the genomic distance, their interaction profile across the whole chromosomes is very similar. Right? So what does it tell us? Right? How do we analyze this data? Right? Again, I think you have covered the PCA analysis, or you will cover PCA analysis. Right? Essentially, PCA just gave you the dimension reduction. Right? So in this case, we can use PCA analysis to reduce this high C matrix into this 1D eigenvalues, eigenvectors. Here, just what we show is only PC1, right? So for PC1, you can see the positive values and negative values. Amazingly, you can see the PC1 values and the positive and negative values. They are really corresponding to the checkerboard patterns in the genome, right? And when the authors, they compare the eigenvectors with the gene density, with the homomorphism marks, they observe actually the positive values, which they call compartment A, is really corresponding to the gene dense regions and a lot of the active transcription values. And the negative values for eigenvectors, they really corresponding to the gene desert. And a lot of times they're enriched for repressive marks. So one thing you can observe that actually potentially the A compartments, right, they still interacting with A compartment, even if they are pretty far away from, they are pretty far away in terms of the genomic distance. Right? And the B blue part time, the blue bars, they are also interacting with the blue regions. A interacting with A, and B interacting with B. So what, again, we are talking about cells. What does it really tell you, right? Again, so this is a very nice review written by Jaap Dekker last year. Again, in the beginning, you have your cell. This is cell nucleus, different chromosomes or chromosome territories. And essentially A and B. A is more active, means they are relatively closer to the inside of the cell. And compartment B, they are closer to the outside, to the nuclear envelope, to the lamina, meaning B is more repressed, right? And the idea is because all the A's are really inside the nucleus, they tend to interact, because once you imagine the cells are folded, so A are more in the inside, so the A tend to interact with A compartment, even if they are pretty far away from each other. And B is similar story. So that's why you begin to see these checkerboard kind of patterns, right? Again, only we, or the authors in that paper, they only define A and B compartment essentially based on strong positive and negative values, right? Which is good. But as a mathematician and a computer biologist, you can imagine, oh, sometimes there's strong enrichment and sometimes there's really, really low negative values, right? Maybe you can put them into a different degrade, right? So further on, I think there's a Rao, ETL 2014, and a few following up papers, they began to further dissect the A and B compartment to subcompartment. So they began to say, okay, there's, besides A and B, there's A1 and A2 compartment. There's a B1, B2, B3 compartment. The potential hypothesis actually A1, A2, and B1, 3, B2, they are interacting with different partners, right? So in this case, you can see, again, A is more in the center, and those regions are more open and then more transcriptionally active, right? So you can see in A, they are the compartment closer to the nuclear speckles. There are a lot of transcription going on. And A2, they are more PAL2 and Bruno domains in this region. Again, but both A2, A1, A2, they are open compartment and actively transcribed region. So for B, because there are different repressive mechanisms, right? For B1, it has been proposed, there are a lot of the polycomb proteins. There are many polycomb proteins, they come together, sort of like have their own phase separation kind of mechanisms, and all the DNAs will come over surrounding those polycombs and really repress the gene expression. And the B2, they are pretty close to the nucleosis. B3 is more on the edge. They are closer to the lamina, and HP1, they're enriched for HP1 protein, right? Again, to further dissecting the genome into different compartments is still an actively studied field. And there's still space for you guys to do some damage in that field as well. Okay, so previously we talked about tags are pretty consistent. So how about compartment, right? It's quite amazing. In this case, again, we are talking about the same 5 cell types, ESL, and the 4 different cell types. Again, if you only see the tags or the high C interactions, they are pretty much the same, right? But once you do your conversion, you calculate their eigenvectors, you can see their compartment is quite different. You can see these 5 cell lines, high C are very similar, but compartment, you can see There's a B compartment, A compartment, B compartment. It's quite divergent. So it seems like if the compartment annotation, or at least the PC1 eigenvectors, they possess more diversity, right? And then you may ask, so whether the A and B compartment, they will have any effect on the gene regulation, right? I will use one example in our lab. Uh, in this work, all right, we have two conditions. Condition one, condition two, where we did CRISPR, right? So you can observe in this case, this is a B compartment, this is A compartment. And these two purple tracks are the ChIP-seq for H3K27 isolation, right? And then the last part is RNA-seq. What we're investigating is called, it's a leukemia gene, PON2 gene, right? So in the normal condition, this region is B compartment. But once we did our CRISPR experiment, this region becomes the A compartment. And as a result, the expression for this gene is upregulated by almost 5-fold, right? And then when we further compare what we observed in the cell line in the patient, again, so this is Hi-C data. In this case, you can see these few guys, this patient and cell lines, they are A compartment, and these are B compartment. Again, we observe this gene is highly expressed in the A compartment rather compared with the B compartment. And overall, when you manipulate the genome, you can, in general, the genes, when they are located in the B to A switch, their gene expression is really upregulated. And when the genes are located in the A to B compartment, their gene expression is really reduced on a global level, right? Okay, so for the next topic, we will begin to look at the Crompton loops, right? So those are more potentially more related with the gene regulation that you are familiar with, right? Again, so what we talk about, Crompton loops is actually here. You can see when there are two parts of genome, they are contacting each other. Potentially one part is a promoter, the other is an enhancer, right? When reflecting on the Hi-C map, it will be represented as this red bright dot in the genome. So again, that's a field you can make your contribution, right? Looking for the better algorithms to identify such dots on the Hi-C map. So this is a very nice review written by Schmidt and Minghu and Bingren's group. So essentially, they list quite a few commonly used software to call Crompton loops, right? So essentially, I think right now, probably Hiccup and FitHiC is a little bit more widely used in this field because Hiccup actually was initially released together with this Hi-C paper from the same group, Aris Lieberman's group. So either, I think all the software in that table, essentially, they just compute a global expected value, and then they will looking for the enrichment, and further they will also compare and looking for the strong local enrichment. More or less, their approach are like that. So last year, actually, there's another group publishes a good paper. Pikachu happened to be my group, right? So in that paper, we began to use a machine learning kind of approach. The idea is whatever you observe here, you are only looking for the patterns. But if you can give me a set of positive values, give me the known interactions, then we can really take advantage of supervised learning and then to predict the loops genome-wide, right? And in this case, we're able to use two different sets. They are from different technologies that I will introduce in about 10 minutes or so. The idea is you train with different input data sets, you can make predictions. So you can see this is actually in the GM12878 using different training data sets. We are able to catch some really nice looking Crompton loops. And then once it will dissect what is enriched at the two loop anchors, you can see most of them, they are enriched between enhancers to promoters and enhancers, and sometimes promoters to promoters. So there has been some research recently began to show that one gene's promoter can potentially act as an enhancer for another gene through Crompton looping. Again, that's a very interesting observations, and I believe more paper will come out on that direction. Okay. And then you can use some of your homework assessment. Again, you are looking for what kind of transcription factors or his modifications are enriched at loop anchors. So this is the paper we did in, this is the result from our paper. We collect all the quantum looping events and then we did the motif search. Not surprisingly, we see CTCF and then two unit of the cohesion complex, SMC3 and Rad21. We see the enrichment of these three guys, which is quite reassuring. And we also began to see ZF143 and then YY1. Interesting, these two factors has been also been tested to be important for the formation of quantum loops. People have done CRISPR experiment to show the function for these two factors. And then you can see there are also enrichment for other factors, including potential TFs and His modifications. So if you are interested, I think there's a lot more for you to study. All right. And then people usually ask, when you do Hi-C analysis, what's the sequence depth you need? For Lieberman's cell 2014, they actually sequenced close to 5 billion reads, right? So usually you need to have billions of reads to reach a 5 kb resolution to call loops. So one of the contributions we made with this Pikachu software is actually because it's a supervised learning, and in the process, we're able to give you essentially the same set of prediction, only use 50% of reads. And then when you go to extreme, we only use 1.5% of reads, which are corresponding to about 30 million reads. You can still get a set of a close 1 or 2,000 high quality loops, which really gives us hope that potentially as a statistician or this young bright students in this group, maybe you can write softwares to really reduce the requirement of a deep sequencing for Hi-C data analysis. All right. Again, once you identify quantum looping events, you can begin to identify cell type specific loops. In this case, again, this is a paper in bioRxiv. In this work, we performed Hi-C in two different subtype of bladder cancer. So one's called luminal, which the other calls basal. So essentially, that's I think they name it using the similar genes to breast cancer. You can see the first two groups are more like luminal subtypes, cell lines. These two parts are more like a basal, right? So through Hi-C data analysis, you can see the enrichment of a subtype specific loops, right? I will just give you one example. In this case, the first two track, so these are the luminal specific cancer. The next two tracks are basal, right? So we can see these luminal specific quantum looping events, they really link the luminal specific His modifications and potentially regulate FOXY1. I'm pretty sure Shirley is familiar with this gene as well, which is one of the luminal marker genes in multiple type of cancer, right? And this shows you a type of basal specific loops linking, again, this loop only observed in Hi-C data in basal Hi-C and linking basal specific quantum His modifications. All right, so I think I'm going slower than I expected. All right, I'll do my best. Okay, so again, so far we have been all the observations have really made using Hi-C data, and then you can see there are different variations of the so-called C-based technology. Again, the name might be a little bit confusing, but let's see. 3C, it's 1-to-1, right? 4C essentially is as it can give you 1-to-all. So you just need to set up one gene as your anchor. You can look at genome-wide which region is interacting with your anchor point. Of course, Hi-C is all-to-all. There's also capture Hi-C and the Chia-PET. I probably will just go over two of them. So capture Hi-C, essentially, you can imagine this as an extra step, right? You can see this is actually published in Nature Genetics 2015. All you need to do is first you construct a Hi-C library. But because, again, we said Hi-C, you need to have 5 billion reads to give you the potential 5 kb resolution, right? Sometimes people say, oh, I really don't need to know all-to-all. I only care about gene promoters or certain regions. In this case, you can just design several probes. You just capture those probes. Essentially, you're enriching for the fragments that are interacting with your target regions, either promoter sets or enhancer sets, right? So Chia-PET or HiChip, they are essentially, the design is similar. Essentially, you have your Hi-C library. This is a cross-link. Again, different quantum looping events. they are being tethered by different proteins. So in the previous capsid, you are enriched for a probe. In this case, they are just looking for the regions uh, uh, only directed by certain proteins. In this case, you can, before you do the final biotin pull down, you can pull down, just like ChIP-seq, you pull down your Hi-C library for specific proteins, and then you do finish your Hi-C experiment. In this case, you can see the Hi-ChIP libraries and Hi-C globalized wise, they are really similar to each other. And then when you see at local regions, potentially the coverage for a whole region is a little bit thin. But the beauty for ChIP-PET and capture Hi-C is actually it only requires, say, 60 to 100 million reads rather than billions of reads, right? But those are enrichment-based assay and won't give you complete genome-wide all-to-all interactions. All right. So 4C again is one-to-all. Essentially, you can imagine this is more like just one row on your Hi-C data. You have an anchor, and you just want to see in this whole region which part is interacting with your anchor region. Right? So once you have the bump like this, that tells you, okay, so these two regions are interacting to each other. Okay. Okay. Again, different technologies. So I think again that you can have that a cheat table to tell you the difference. But to me, all right. There are actually an ongoing effort to comparing the technologies between all the way from 4C to Hi-C. Right? Again, I probably will use 10 minutes or so to introduce other technologies. I hope I don't overwhelm you with this. This is just a good story. I think I just cannot stop. All right. So far, a key point for all the 3C, 4C experiment is actually once you have your DNA cut, you cut a DNA, you need to ligate your DNA. You have these 2 points, and then you do a PCR, you do your sequencing library or capture, right? Just ligation. So potentially the cutting and the ligation efficiency will have a direct effect on your libraries. So the other thing is when you're doing this ligation events, essentially you are looking at this looping event, just one loop, right? And then there are some other 3D genome structures potentially you might miss based on the ligation events. So this is called a genome architecture mapping technology, or GAM, was published in Nature 2017. I think the design is very smart. So essentially, this is your cell. You just slice the cell, take a piece of the cell, right? And then imagine there are 2 loci. You take one cut. In this case, red and green, they're on the same slice, right? In another cell, you do the same slice. In this case, red and green are together, but the blue fell off. You do more and more slice in different cells, even the same cell, you do multiple cut. As a result, if A and C, they are consistently on the same slice, that means potentially they are really close to each other, right? Because you cut the cell in different angles, in different cells. So similarly, they can just construct a Hi-C-like matrix, and they can give you the potential interactions between 2 parts. Another technology is called a Sprite, which is invented by Mitch Gutman. So in this case, I know you haven't covered the single cell yet, but this is actually sort of like a split pool you might be familiar with later. Essentially, you just put your libraries into different 96 wells, right? So of course, you can imagine different complex or different looping events. They might fall into the same well. You give the barcode, so you barcode these 2 complex, like orange, right? And then these, well, they have different color. And then you get them out, split, and put them back in the well, right? The idea is for the 2 complex fall into the same cell with the same barcode is really slim, right? And then you keep doing this split and pull, split pull. In the end, 2 complex have exactly the same assignment of the well in different colors. It's quite small, right? And then in this idea, you can actually sequence your libraries. Again, you can compute a nice library like this. Again, the advantage of this no ligation-based technology over Hi-C is actually they don't require this looping, because sometimes you have big protein, they can actually have multiple DNAs attached to it. These can give you the complex events. There are multiple DNAs for a same complex. They don't need to interact with each other, but still, this technology can capture them in the experiments. Right? So there are similar technologies called ChIP drop. they are taking the 10x genomics assay. So essentially the same idea. Each complex is captured by this oil bubble, and then each complex will have their unique barcode, and you sequence your library. All the reads in the same complex will have the same barcode. And again, you assemble them together, put them in a Hi-C like map. Again, like a GM, like Sprite and SharePad, they are potentially better at detecting the complex events where have multiple N come to the same region rather than one-to-one looping events. All right. Okay, in the next 10 minutes or so, I will going to share with you some of the tools that I really like and that they are widely used in this community, and potentially they might be helpful for you to explore data and use it for your own research. So once you have a Hi-C data library sequenced, what do you do, right? A really popular tool that you can use in your desktop is called Juicebox. It's still from Lieberman's group. The idea is actually you can use this app. You just download Juicebox, and then once you generate your Hi-C library, you process it. You have your Hi-C file. You can load your Hi-C with Juicebox, and you can just double click and zoom in, zoom out. It's really fast, right? You can also see the inter and intra chromosomal interactions. They can also do the eigenvector value confirmation for you. I really like this tool. You can use it. You can potentially also add the one-dimensional tracks for you as well, so they can link different part of genome with the quantum looping event to ChIP-seq signals. So another really great resource is WashU Epic Genome Browser. Again, it's invented by my close friend Ting Wang from WashU. So the advantage for WashU Epic Genome Browser is actually beyond. So the Juicebox, that's a application. I think they began to also have a web application as well. But the WashU browser is really a browser based. It holds more than 10,000 genomic data sets from ENCODE and roadmap and lots of Hi-C data. The idea is actually you can load your Hi-C matrix, you can manipulate the heat map, and then they also have this arc-like structure. Once you give the predicted looping events between the two loci, you can align them with these tens of thousands of tracks together. It's really a great resource for you to play with, right? And then another great web resource actually was invented by my group. This is called the 3D Genome Browser. This browser actually, we just checked, has been used for more than 1 million visits in the past few years from over 100 countries all over the world. So the idea is similar. We have preloaded, everything's preloaded. The annotations, I don't keep all the tracks of ChIP-seq raw signals, but we do have also their peak files. And then we preloaded more than 300 genomic Hi-C or captured Hi-C type of 3D genome data sets. And then all you need to do is just go to the website. It's 3dgenome.org. I hope it can appear on the screen. 3dgenome.org. You can check on your iPhone. You can use it on your computer. All you need to do is go to the website. It's specifically designed for Hi-C and type of 3D genome structure. You can just choose your cell type from the drop-down menu and type your gene, type your GWAS, type your genome loci. That's it, right? You just go there, choose your genomic regions, click submit, and you will have something like this. We can show you the Hi-C map. You can see this is a quantum interaction between this region to the other region. Right? So for this 3D genome browser, you can also show multiple tracks as well. So in this case, you can see, you can even load different tracks. In this case, we're showing the human and the mouse for the similar gene. You can see the conservation of the tag structures. And we can also host built-in session for both Epic Genome Browser and UCSC Genome Browser, so that it can, again, align your ChIP-seq tracks with the Hi-C data matrix. Again, so far we have loaded 30, 40 Hi-C paper data sets, more than 300 of them. Every data is processed using the same pipeline. Loops are called, use Pikachu, and tags are also called with our DI models. So you are more than welcome to come to our website and use this resource. Again, so one of the community I really have to stress is this NH40 nucleosome projects. It started about 5 years ago, and the phase 2 just started last December, right? So for this consortium, We have more than, I think, 30, 40 investigators from all over the world, and people are studying the 3D genome structures using both the genomics assay and a lot of imaging-based assay. And the idea is that people will compare different genome assay itself and also compare the observations you can make from genomics with imaging. The idea is we'll see if we can have a really a great complete picture of the 3D genome. Of course, 4D here means really you also have another state, 3D plus time series, right? During differentiation or during health to disease transition. So that's where the 4D come from. So again, the 4D 4D has processed a lot of data. Many data are deposited to the data portal. They also have processed a lot of the Hi-C and RNA-FISH and the TSA-seq different data sets, right? You can go to the 4D portal. If you just search 4D portal, you can see the data. Again, all the data, you can download them. They are, DCC is funded by the 4DN, so they have a lot of resource. All the raw files and the matrix and then the BAM files can be downloaded. So this is a very nice visualization as well. So again, you can go to 4DN to download a lot of the pre-processed file as well. It's a great resource, right? All right, to summarize today's lecture, I know I gave you a lot of terminologies and a lot of the technologies and concepts. This is something I think might be really important for you for your future research. 3D genome is really tightly linked with the gene regulation, right? And currently there are two different main approach: is imaging-based versus a genomics-based. And even for the genomics-based, there's a ligation-based technologies, different C technologies versus non-ligation-based technologies. And of course, compartment A and B, they're enriched for different chromatin compartment, and for example, U chromatins and the heterochromatins. And then TADs, they're conserved between different tissues and species. And then the disruption of TAD boundaries might have a severe effect in the phenotype and the gene regulation. And personally, I think that the importance for the test is really they are preventing the wrong wearing between the enhancers or even the repressors to their target gene. And then we also show you that you can use software to detect dynamic quantum looping events and to their target genes. And then one thing we really don't have time to talk about, there's again, single cell Hi-C technology is also emerging. And then if you're interested, I think when Shirley covered the single cell attacks in R6, you can do more research on the single cell Hi-C as well.